This is the flip lesson on the continuation of the organelles that we've been studying. And one thing that you need to know is the jelly-like fluid, I guess you could call it, inside the cell that suspends everything. So if you can imagine making a bowl of Jello, and then maybe you put some things in the Jello, like let's say you put gummy bears in the Jello, and then those gummy bears get suspended in it as it thickens and hardens, and it becomes Jello. So imagine those gummy bears in there. That's kind of analogous to the organelles that are in a cell. Okay, so these organelles that are in the cell that we've talked about so far, we've talked about the mitochondria and we've talked about the nucleus. Those are suspended in this fluid-like medium called cytoplasm. Okay, so cytoplasm here, the structure of it pretty much is just a fluid. Okay, and then the function of it, again, structure and function are key. It suspends the organelles, okay, or it kind of like floats them around. It keeps them functional so that they could go about what their jobs are, okay? If there isn't cytoplasm in there, then the organelles wouldn't be able to function, okay? Because they need to be suspended within this fluid-like medium, okay? So that's the cytoplasm. I'm also going to do the plasma membrane. Now the plasma membrane is called many different things. So it could be called the plasma membrane. It could also be called the cell membrane. And then another name for it is the phospholipid bilayer. So you could actually use these three terms interchangeably. They all mean the same thing. The structure of the plasma membrane is double. That's why this prefix here, you may have heard that like mono is 1, bi is 2, okay? So this bilayer, what that means is double layer. And it's a double layer of phospholipids, okay? And these phospholipids look like bunny heads with the ears attached. So here's one phospholipid, here's the other phospholipid, okay? Now a cell doesn't just have two phospholipids like this, it actually has like a ton of these. And these are like the barrier that exists between the cell's internal environment and the cell's external environment. So what I just drew there is basically a magnification of this. So what I've done is I've magnified this in a sense. So here's your nucleus on the inside, right? And we've also talked about the mitochondria, that would be right here. And cells, by the way, could have many mitochondria. It just depends on how much energy is needed for wherever the cell is. Okay? So for example, if you were to take a sample of a heart cell and count the number of mitochondria in a heart cell, there's like a ton more there than you would find in a cell maybe from the tissues in your big toe. Okay? And that's why. Why do you think that is? Well, it's because your heart's always working. It needs energy. So remember, the mitochondria is the powerhouse, right? It provides that energy. So you're going to find a ton more mitochondria in a heart cell than a toe cell, right? So with that said, something that they have in common between a toe cell and a heart cell is the phospholipid bilayer. They both have this barrier, right? This barrier that separates the cell's internal environment from the external environment. Okay? So the structure of the plasma membrane, we need to know, is a double layer. Okay? And it's a double layer of these here. These are called phospholipids. This part of the phospholipid is called the head, and then this part is called the tail. Okay? And what you would find is that if this was magnified here, this would be like the outside of the cell. And then on this side would be the inside of the cell. So hopefully you're oriented right that way. I'm not confusing you. So inside would be here with where I wrote that word, and then outside would be here where I wrote that word. Okay? Now the function, again, remember we talk about the structure and the function. The function of the phospholipid bilayer is that it basically selects what enters and exits the cell. Okay? It chooses what can come in and it chooses what can go out. So it's kind of like if you've ever been to the airport, like the TSA. They only allow certain things in and then they do not allow other things, right? So it's kind of like a bouncer or a barrier or a filter is another word for it. So it controls, the function of it is it controls what enters and exits the cell. Okay? And in a few weeks, we're going to learn that it's just not this simple. This bilayer here, you guys, has like a ton of things embedded in it, which is like what this stuff is here, but you don't need to write any of this right now, okay? Because we'll learn about that later. But what I do want you to know and just keep in mind is that this phospholipid and this phospholipid, it's not just phospholipids all the way around. You can see that there's this thing here, this thing here, this thing here. So there's a bunch of other things embedded within it that help with that control right here, okay? So there's other things embedded, we'll get into later on, that help to control what enters and exits the cell, okay? All right, and that's it for cytoplasm and plasma membrane.